questions which have been coming primarily in the context of Mendelism, sex linkage and genetic abnormalities run into different frames. Like you see the first question, what contributes to the success of Mendel? Is it qualitative analysis, observation of distinct phenotypes, knowledge of biology, or consideration of one character at a time? Consideration of one character at a time is the right answer because it is either tallness or dwarfness, round seeds or wrinkled seeds, or axillary flowers or terminal flowers. So, taking a character at a time when taking a pair of characters is a secret towards the success of Mendelism. The second question says, what is the diploid number of chromosomes in humans? The diploid number of chromosomes is given as 46. That is, we have 23 pairs of chromosomes. 23 is inherited from the paternal side and 23 from the maternal side, and together they are termed as the sort of an autosomes. So, the diploid number of any particular organism is constant, the chromosome number is constant, the chromosome number is the basis for inheritance, and chromosomes are the vehicles of inheritance. The third question says, what happens if there is a mating between a capital T, small t into a small t and a small t? What will be the characteristics of the offsprings? So, if you take them as the parents, from this side we have the gametes, you have a capital T rounded and a small t rounded, and similarly we have a small t from the right hand side. A crossing between them, probabilistic crossing, we have a capital T and a small t here, and a small t and a small t here. Small t and a small t here, we find that 50% of them are tall, heterozygous, and 50% of them are dwarf homozygous. So, as per the question, the characteristics of the offspring is they are 50% recessive. Now, we are going on question number 4. In question number 4, it is asked, what is the cross between a capital A, small a, capital B and a small b, and small aa, small bb. So, a cross between these two parents, if they are taken as the parents, we have from one side we have a gamete AB rounded, A and B rounded, then we have a small a and a small b rounded, and from this side we have a small a and a small b rounded, and a combination of these, you have a capital A from here, you have a small a from here, you have a capital B from here, and a small b from here. One of the gametes is capital A, capital B and small b. Another of the gametes is small a, capital B and small b. These are the two important gametes which get formed over here. Thereby, the choice which is the right answer for A says the answer is 1 capital A, capital B and small b is to 1 small aa and small bb. This is with relation to the fourth question, and the right answer is the A bit. The question number is 5. In 5, it's asked, in Down syndrome of the male child, what is the sex complement? As implied in the case of the male child, it is always XY in the case of the Down syndrome. While we had studied on Down syndrome, you had known that the answer is XY. Hence, the correct answer for the choice answer is B, is XY. As red antiharem is crossed with white antiharem, which are both homozygous. The offsprings are RW is white. So, this is an example of incomplete dominance. So, in incomplete dominance, there is an expression of both the genes and both the genes will blend as seen in RW, which is pink. Hence, the answer is B. So, this is an example of incomplete dominance. The seventh question asks, what is the dihybrid ratio? A dihybrid ratio is predominantly the condition is seen as a heterozygous condition. Heterozygous condition of two genes which are inherited together to determine a genotype as well as a phenotype. So, when you are looking at a particular sort of a character which is determined by two genes, and you are looking at their inheritance pattern, and it is seen in the heterozygous condition, the correct answer is you have a capital T and a small t and a capital R and a small r, which determines that it is a heterozygous condition. Hence, the answer is D. Eighth question asks you, who had been the first genetist, or who has been the father of genetics? The father of genetics, as you understand, has always been Mendel. Mendel is associated with classical genetics. And he was the first scientist to propose about transmission genetics. Transmission genetics is a study of inheritance. That is how characters are transmitted from the parents to the offspring and variations thereof. Whereas, if you take De Vries is associated with mutations and polyploidy. Darwin, the next choice, relates to evolution, and Morgan is associated with sex linkage and sexual patterns of inheritance, or the chromosome pattern of inheritance. So, the chromosomes chromosomal inheritance as a basis has discovered by Morgan. Morgan had primarily worked with Drosophila or Drosophila genetics, whereas Mendel had worked with Pisum sativum, and De Vries and Darwin both are associated with evolution and even a bit of mutation. So, the correct answer to question 8 is B, that is Mendel. In a Mendel's cross, when we take question number 9, we are looking at Mendelian crosses. When we have Mendelian crosses, we deal with factors or genes, and they are of contrasting pairs of characters. We have a contrasting pair of characters, like you have a capital A, small a, capital B, and a small b. And the contrasting pairs of factors or the crosses are predominantly termed as allelomorphs, morphologically which are different, but which are determined by a pair of genes which are termed as allelomorphs, whereas multiple alleles. So, the correct answer is B, which refers to allelomorphs. While if you take multiple alleles refers to a cluster of alleles which have got a common point, and alloloci and paramorphs are out of context in the objective bit. The 10th question talks about multiple alleles. Multiple alleles, as you understand, are the basis for the control of inheritance in A, B, O blood groups, which I have already studied. So, hence the answer is D. While if you take sickle cell anemia, it is because of a single gene. Same as the case with relation to color blindness. And phenylketonuria is a metabolic disorder which is not inherited. So, the correct answer is D, related to blood groups. So, we go to the 11th question. The 11th question says a colorblind girl is rare. A colorblind girl is extremely rare, and she'll only be born if her father and maternal, that is maternal grandfather is colorblind. The answer to the 11th question is B. The reasons being, father is a carrier for color blindness. He has the gene for color blindness. The mother, the maternal side, are the carriers. They are the carrier for color blindness, and maternal grandfather has again got the colorblind gene. Though phenotypically, as we understand, whether it is the father or the mother or the grandfather or the girl, phenotypically they all are similar, whereas the chances of a colorblind girl is extremely rare, predominantly because the girl is a sort of a carrier, is a carrier of color blindness, and she is a carrier and she inherits the character of color blindness from her mother. And mother is also a sort of a carrier because she inherits the gene from the grandfather. So, the color blindness gene from the grandfather goes to the mother, and from the mother the carrier of color blindness is a sort of a girl. So, hence the probability or the answer to question number 11 is B. Next we go to the 12th question. The 12th question says an organism with two identical alleles is predominantly termed as either a dominant, hybrid, heterozygous or homozygous. So, the 12th question, the answer is homozygous. The homozygous condition indicates when an allele which is indicated as a sort of a gene with a dominant and a recessive trait at one single locus, and which is similar, responsible for the formation of a phenotype, a typical phenotype. So, when we are dealing with a gene and which have got similar phenotypic characters, and either they could be either recessive or they could be dominant. However, they are present on a single locus. The morphological expression of a trait genetically, which is identical and expressed on a single gene, a single locus. It is identical phenotype. It is predominantly termed as homozygous condition. We refer to this as homozygous condition. Hence, the answer for 12 is D. While if you take dominant, dominant is the expression of a particular character wherein one particular character remains expressed and another is hidden. A hybrid is normally possible in an F1. In an F1, we have the characters of both the parents as well as the offspring. And a heterozygous condition predominantly indicates a capital A, a small a, capital B, and a small b. Whereas in a homozygous condition, it can be represented as AA is homozygous dominant, or it can be BB homozygous recessive. Otherwise, it could be capital A, small b, which again indicates a sort of a homozygous condition. Otherwise, it can be a small a, and a capital B, which is also a homozygous condition. So, there are 4 possibilities. We have AA as a possibility, BB as a possibility, capital A, small bb as a possibility, and again small a and capital BB as a possibility, which are all examples of the sort of a homozygous condition. Next, we go to the 13th problem. In the 13th problem, it is asked, in a cross between capital AABB into, we have small AABB, they are asking what is the ratio of the F2 genotypes. The ratio of the F2 genotypes. What is the ratio of the F2 genotypes? Typically, in case we have a capital A, small a, capital B, and a small b, as well as if there is a capital A, and a small b, and a small b, and again you have a small b, and a small b. And primarily here, when you take this particular thing, the ratio, the answer to the ratio is given as 1:2:2:1. It's given as 1:2:2:1. We need to do some deduction. So, this is deductive genetics. So, from here we have the gametes. We have a capital A and a capital B coming from here, which is a gamete. And again we have a small a and a small b coming as a sort of a gamete. So, these are the parents, these are the sort of a gametes, and when you take the F1, in the F1 it will be a capital A from here, and again you have a small a from here, you have a capital B from here, and you have a small b from here. So, a typical F1 is heterozygous for a capital A, small a, capital B, and a small b. Now, we are looking at F2 genotypes, that is F1 into F1, that is which is selfed. Thereby, we have a capital A, capital B, small b into a capital A, capital B, and a small b. This is termed as F2 analysis, or it is F1 into F1. Otherwise, it is also termed as selfing. So, when you take this sort of a combination, we understand we have a capital AB gamete on one side, then we have a capital A, small b gamete on the other side, then again we have a small a and a capital B, and again we have a small a and a small b. So, this is the typical sort of a combination which occurs in the gametes. And same is true with relation to the other side. We have a capital AB, we have a capital A small b, and we have a small a capital B, and we have a small b. Now the question says, what exactly is the ratio of the F2 genotypes? The ratio of the F2 genotypes is, if we take the combination here is AB and AB comes once, so this indicates 1. And again, when you take a small AB and you take another small AB, this ratio is again as 1. This is indicated as 1 and 1, and the rest of the combinations, whatever it occurs over here, these are 2, 3, 4, 5, 6. So these are 2 and 2. Hence the answer is 1:2:2:1. This is problem number 14. If an allele, that is a pair of contrasting characters which is represented by alphabets in the genetic notation, is dominant, if an allele is dominant in, it should be both in the homozygous state, that is as capital A, capital B, or as small a, small b, as well as it should also be dominant in the heterozygous state. A simple example of this particular logic is, you think we have capital A dash B dash, it should be 9, capital A dash small bb should be 3, small a capital B dash should be 3, small a small bb is 1. Now when we take dominant alleles are normally seen when in this condition it is dominant, as well as even in this condition also it is dominant, even in the third condition it is dominant. So dominance primarily refers to whether it is in the homozygous condition or in the heterozygous condition. So any allele is, you call it as dominant when it can be this way. You take the situation of capital A dash B dash. Capital A dash B dash can be AA, or it can be A and small a, as well as capital A dash B dash can be capital BB, or it can be capital A dash B dash can also be small b. So there could be different sort of combinations. So the allele for dominance, the correct answer is predominantly it is A. So for the 14th question, the answer is A, which indicates dominance which is present both in the homozygous as well as in the heterozygous condition. Now we move to the 15th question. The 15th question says, in human beings, if we have got 45 chromosomes, that is, we have 23 pairs of chromosomes, that is, you should normally have 46, but however, if you got 45 chromosomes with a single X, or it could also be X and O. O means Y is missing. Y is missing. This sort of an abnormality is predominantly seen in Turner syndrome, as we have already discussed about this. So in the case of the Turner syndrome, we have a loss with only there is presence of a single X with a 45 chromosome. This is an attribute of Turner syndrome, which is given as the correct answer is C. We are going to the Down syndrome. So in the Down syndrome, we understand that this sort of a syndrome, as you understand, is because of trisomy of chromosome 16. Here in this situation, the pair of chromosomes on chromosome 16 do not separate, or they do not segregate. during cell division. During cell division, whether it is mitosis or meiosis, non-separation of the chromosomes during cell division is termed as non-disjunction. Hence, the answer for 16 is, the answer is D. That is the non-disjunction of the chromosomes. So, non-separation of chromosomes due to blockers, due to blocking, due to an abnormal sort of a chromosome, a piece coming and interfering with mitosis and meiosis is predominantly termed as non-disjunction. This is the case in the case of the Down syndrome. In the case of the Down syndrome. We are going to question number 17. A colorblind mother and a normal father would have what sort of sons and what sort of daughters? Option number 1 says presence of colorblind sons and normal carrier daughters. Option number B says colorblind sons and daughters. C says all are colorblind. D says everything is normal. Now, you exactly understand when you take the mother. The mother here is always colorblind carrier. Colorblind carrier. And when we take the father here, the father is normal, that is without color blindness. As implied or studied in the concept base, we always find that the sons are colorblind, and the daughters are the carriers of the colorblind gene, though both of them show normal phenotype. Hence, the answer for the 17th question, the answer is A. Now we are going to the 18th question. When a certain character is inherited only through the female parent, it probably represents A is multiple plastid inheritance, B is cytoplasmic inheritance, C is incomplete inheritance, or D Mendelian nuclear inheritance. Now, in plants, when we are dealing with inheritance, several characters like sterility, cytoplasmic male sterility, are inherited through the cytoplasm. It means the maternal cytoplasm carries the genes for inheritance. So, the maternal cytoplasm includes the chloroplast as well as the mitochondria. Chloroplast and mitochondria are otherwise termed as organelles. The chloroplast and mitochondria have got their own chloroplast DNA and their mitochondrial DNA. So, a pattern of inheritance which is determined by the cytoplasm or the maternal line or through plastid inheritance and is termed as cytoplasmic male sterile. So, during cytoplasmic male sterility, we have the encoding of the gene for ribulose diphosphate carboxylase in plants. And similarly, if you take in the case of Chlamydomonas, there are peptide particles, which are also seen in the case of Euglena. So all of these are examples of cytoplasmic inheritance or maternal inheritance, and they are the genes for male sterility. Hence, for the 18th question, like if it is inherited through the female parent or through the egg or the cytoplasm of the egg, the correct answer is B, that is cytoplasmic inheritance. Cytoplasmic inheritance. While the other choices, incomplete dominance indicates when two genes come from both the parents. Mendelian nuclear inheritance deals directly with dominance and recessive, and multiple placid inheritance is a complex situation. 19th question says, of a normal couple, half the sons are hemophilic while half the daughters are carriers. The gene is located where? Now, as you understand, if you take hemophilia, hemophilia is a bleeder's disease. And hemophilia is sex linked. It is sex linked, and normally it is transmitted from the mother. It is present in the father. The gene is present in the father, but mothers are the carriers, and it is given to the sort of a daughter. So again, the daughter is also the sort of a carrier. Daughter is also the sort of a carrier. So when you say the mother, it means on one of the X chromosomes, of the mother. Hence, the answer is C. Answer is C. So we understand that it is sex linked. The gene is represented on the father. It is mothers are the carriers. Daughters too are the carriers. It is located on the X chromosome of the mother. We go to question number 20. Mr. Kapoor has capital B small b autosomal gene pair and D allele which is sex linked. What shall be the proportion of capital B small d in the sperms? The choices are A is 0, B is half 1 by 2, C is 1 by 4 or quarter, D is 1 by 8. So when we have capital B and small b, which is autosomal gene pair, and we have the D allele which is sex linked, given that the parent is capital B and small b and the sex linked gene is capital D, small d is sex linked, the proportion of capital B and D in the sperms among the choices is we have is 1 by 4 or 25%. Hence, the answer is C. It is a study of color blindness. In color blindness, the chances of a male child being colorblind. The question says of both normal parents. We have got two parents who are normal. We have a normal male and a female, and we are looking at the offspring. We are looking at the male child. The chances of the male child bearing colorblind is only when the mother's father is colorblind. Supposing you have the mother would always be the carrier, and the mother's father would be colorblind because her husband is normal. So if they inherit and if the son is colorblind, the colorblind gene of the son comes via the carrier of the mother from the father who is colorblind. So the transmission occurs from the grandfather of the son to the mother and to the son of a son. So the possibility exists when both the parents are normal. when both the parents are normal, if the male child has to be colorblind, the possible only when the mother's father was colorblind. Hence, the answer is D. Correct answer is D. The question number 22 says, a woman with albinic father. We have a woman with an albinic father. The father is albinic. Albinic father marries a man who is also albinic. The proportion of the progeny is, the question is asked us, how is the sort of a progeny? The proportion of the progeny is how many are normal and how many are albinic. Now, albinism is sex linked. It is a recessive trait. So, the woman gets the gene from her father, and even the man is albinic. Thereby, the probability occurs as 50% is to 50%. 50% is to 50%, or 1 is normal and 1 is albinic. Hence, the correct answer is D. The correct answer is D. We go to problem number 23. It says, in the problem number 23 says, in a dihybrid cross, a capital A, capital BB is crossed with small a and small bb, and we have the F2. In the F2, they are asking what could be the probable ratio of capital A, capital BB, capital A, capital B, small b, capital A, small b, capital B, and also capital A, small a, capital B, and small b. Now, we represent the typical dihybrid ratio in the form of parents. In a typical dihybrid ratio, when you take the parents are, you got a capital A, capital B, and a capital B, which is crossed with a small aa, small bb. If you take the typical gametes, we have a capital AB rounded and a small ab rounded. And when you take the F1, the F1 has got a capital A, small a, capital B, and small b. This is the notation, how you write the F1. And F1 into F1 is termed as selfing, or it is also termed as F2 analysis. In an F2 analysis, we have a capital AB on one side, capital A and a small b on the other side, small a and a capital B, and a small a and a small b. This is from the Polands. And again, on the X side, we have a capital AB, we have a capital A and a small b, and a small a and a capital B, and a small a and a small b. So, we are looking at what is the probabilistic combination of the gametes, and this is predominantly termed as a checkerboard. So, a checkerboard is a probabilistic device of the combination of gametes in an F2. Now, as according to the question asks, what is the probability of AABB, or how many are AABB, how many are capital A, capital B, and small b, and again how many are capital A, small a, capital B, and capital B, as well as how many are capital A, small a, capital B, and small b. So, we have to seek out answers for this. Now, how do you get a capital A and a capital A? Capital A, and a capital B, capital B. This is a combination which indicates capital A, capital BB. So primarily we have this as 1. We have this as 1, the first one. And predominantly when you look at this, the easy way to know about the key to the diagram is the last, look at the last ratio. It is a capital A, small a, capital B and a small b, or it can be written as this, capital A dash B dash. So when you look at this combinations, these combinations occur over here. 1, 2, 3 and 4. So the quicker a student can understand the 4 combinations here, which are heterozygous, this is an easier way to solve the problem very quickly, wherein you get the answer as D. The answer is D, and the answer is 1:2:2:4. So the correct answer is D, which is 1:2:2:4. And the key to the solution is finding out which is the first one, that is capital AA, capital BB, and the last 4 of the gametes, which indicates the number 4. We are going to the 24th question. The 24th question says a cross between a pure tall pea plant with green pods and dwarf pea plant with yellow pods will produce short F2 plants out of the 16, and what is the right choice? So we are looking at a dihybrid ratio. And the crosses are between a pure tall pea plant with green pods, tall and green. It is pure, homozygous, parents, pure, it is pure, tall and green, which is crossed with dwarf pea plants with yellow pods. Dwarf with yellow, again homozygous. This is dwarf and yellow. As you take the parents, we got the gametes. The gametes are, you got a capital T and a G gamete on one side, and a T and a we have a T and a small g here. Capital T, capital TG and small tg over here. Then we have the F1. In the F1 we have a capital T, small t, capital G and a small g. This is the F1, which is tall, green, heterozygous. The question which is asked here is, how is the combination of the sort of a F2? So we are doing F2 analysis. In F2 analysis, we are taking an F1 and crossing with an F1. So thereby we have capital T, small t, capital G, small g, into a capital T, small t, capital G and a small g. And it is asked how many are short here. So the probability is, here you got a capital TG, you got a capital T and a small g, and again you got a small t and a capital G, and again you got a small t and a small g. You got 4 gametes on either side. So when you get 4 gametes on the either side, short F2 in among the 16 short F2 is, you have is this indicates short, this again indicates short. So 3 plus 1 together makes 4. 3 plus 1 again together makes 4, and the answer here, what remains, is 3. Answer is 3. Hence the choice is B. The choice is B. We go to the 25th question. The 25th question says the genetic identity of a human male is determined by autosomes, nucleus, sex chromosomes or cell organelles. Now, as you understand, autosomes are somatic chromosomes. The choice number A, autosomes is somatic chromosomes. It's not determined by the nucleolus. Nucleolus has got only the chromatin and the RNA synthesis. The correct answer is C. C refers to sex chromosomes. The genetic identity of human male is predominantly determined by the allosomes or the sex chromosomes, and the sex chromosomes could either be XY or XX, which are predominantly termed as the sex chromosomes, or the allosomes, and the last choice is cell organelles. So the correct answer to 22 is C. 26. A person with sex chromosomes XXY suffers from A choice is Down syndrome, B is Klinefelter syndrome, C is Turner syndromes, and gynedomorphism. So normally if there is a genetic abnormality, where there is an X and a Y, there is increase in X, there is an increase in X along with a Y, it is a characteristic feature of Klinefelter syndrome, and hence for 26 the answer is B. Problem number 27 starts. It asks, during Down syndrome, karyotyping has shown that the disorder is associated with trisomy of chromosome 21, and the trisomy of chromosome number 21 is due to choice A is non-distinction during egg cell formation, B is non-distinction during sperm cell formation, C is non-disjunction during formation of egg cells and sperm cells, and D is due to the addition of extra chromosome during mitosis of a zygote. Now we have the situation of a Down syndrome. And in Down syndrome it is trisomy 21. So we have an increase in the chromosome complement. The increase in the chromosome complement is primarily attributed to non-disjunction, that is non-segregation of the chromosomes. Now non-disjunction of the chromosomes here occurs during the formation of both the egg cells and the sperm cells. During both the formation of the egg cells and the sperm cells. So hence the answer to 27 is, we have the answer is C. It involves both egg cells and sperm cells. In the 28th question, it is an example of a trihybrid ratio. How many different types of genetically different gametes will be produced by a heterozygous plant having the genotype capital AA, capital B, small b, and capital C and a small c? So we have a parent which is predominantly heterozygous, and the genotype is given as capital A, capital B and a small b, as well as you got a capital C and a small c. Now to solve this sort of a problem, we have a capital A gene. The capital A gene can again combine with a capital B, or it can also combine with a small b. A can combine with a capital B, as well as A can also combine with a small b. Parallelly, the B can combine with a capital C, or it can combine with a small c. Similarly, small b can combine with a capital C, as well as it can also combine with a small c. When A, B and C combine, we have the formation of a gamete called A, B and C. In the second combination, we have a capital A, capital B and a small c. In the third combination, we have a capital A, we have a small b and a capital C. And in the last combination, we have a capital A, we have a small b and a small c. Thereby, in a genotype of a plant which is capital A, capital B, small b, capital C and a small c, we have the formation of 4 different type of gametes. 4 different type of gametes. Hence, the answer is B. The answer is B.